my research work today that relates to the omega-3 fatty acids and their therapeutic potential in eye pathologies. And my question is it as simple as that? What I'm going to do basically is to have an overview at the beginning of the session. Firstly, I'm going to go through the eye anatomy, very basic eye anatomy and histology, with the emphasis on the retina and the photoreceptors. So those are the most important cells of interest that relates to the visual acuity. And later on, I'm going to go through the different ocular pathologies that might be associated with aging. And the focus will be mainly on age-related macular degeneration and a brief account on Stargardt disease. So this is a genetic condition that relates to similar symptoms with AMD, age-related macular degeneration. In this case, though, this is an inherited condition, and it happens in young individuals. So this is also a condition that is of interest because there is no cure for this type of disease. And then we're following the omega-3 fatty acids and the significance with regards to their function, their role, and how the balance between omega-3 and omega-6 is important in terms of disease progression. So after the brief introduction, I'm going to cover the preclinical studies that we did so far. And the emphasis will be given in two different models. So the first model will be the Black 6 animal model, which relates to normal aging. And the next one relates to retinal degeneration. So we used here a knockout model that mimics the presentation of AMD in humans. After the preclinical studies, I'm going to briefly mention one of the clinical studies that we performed without actually going through the results because the study has not been published yet. But basically, what we did here, we designed a prospective randomized multi-center study where we've seen promising results, and I'm going to only briefly mention that. And to finish off, I'm going to go through the future studies or the future plans that I have in order to perform both preclinical and clinical studies and to expand our knowledge with regards to the omega-3 and how they work in these eye conditions. Right, so just a brief account on the eye anatomy. We know that we have the front of the eye, which is important, but mostly the back of the eye where the retina is found. So I'm going to focus on the back of the eye. But before focusing on that, let's just consider a few basic things. So what happens once we have the light coming in into the eye through the first layer that we see, which is called the cornea. We also have a fluid-filled space behind the cornea, which is called the anterior chamber. And we have the fluid in that area, which is called the aqueous humor. So the aqueous humor is quite important in terms of regulating the the eye pressure of the eye. And this is important for the pathologies of glaucoma. In terms of the back of the anterior chamber, we have the iris. So this is your color part of the eye. And we also have a hole in the iris, which is the pupil. So behind the pupil, we have the lens. So the lens is important in terms of focusing close-up objects while we see for our near sight. Now, if we go at the back of the eye now, we know that the light can come through from the cornea through the lens, and it passes through into the vitreous, which is found in the vitreous body, onto the retina. So the retina, we know that is the light-sensitive tissue that lines the back of the eye. And this is where we actually have the most important cells responsible in order to convert the information into our brain. So I'm going to go through a little bit more about the retina. In terms now of the macula, so this is the macula here, this is responsible for giving us our detailed and central vision. And in terms of the peripheral retina, this is for our peripheral vision. Now, let's focus a little bit on the retina. Why do we need to know about the retina in order to understand later on the studies that we performed? We know that within the retina, which is at the back of the eye, we have the specialized cells, which are called the photoreceptors. So what's their function? The photoreceptors basically change the light into energy, and they transmit this to the brain. And we do have two types of photoreceptors. We have the rods and the cones. So with regards to the rods are responsible for the night vision. So with the rods, you can actually see during the night, and you can see black and white through the rods. Whereas the cones, you have the color vision, and you have more detailed and more central vision with the help of the cones. And we have about a few million of rods. The rods are usually more than the cones. In terms of what happens with regards to from the light, which goes to the retina, we have electrical impulses that they can get into the optic nerve, and they can get transmitted to the visual cortex in our brain. So that's how we perceive an image. So I would like to first go through the different layers of the retina before I actually explain more about the pathologies. We do have a lot of cells, as you can see from the image here, from the diagram. We do have a lot of cell layers that are present in the outer retina and in their inner retina. There are about 10 type of cell layers in the retina. So if we start from the top layer here, this is where we have the choroid. So the choroid basically supplies blood into the outer part of the retina. And after the choroid, we have the retinal pigment epithelium. So this is where we have pigmented cells. And we actually have protection by these cells where they act as a barrier through the inner retina. So they protect the photoreceptors, basically. Now, the photoreceptors are found in the outer nuclear layer. So their cell bodies are found, their nuclei are found in the outer nuclear layer. And the photoreceptors are the ones that are colored in brown, the rods and the cones. So basically, I'm emphasizing this, I'm highlighting this information because the outer nuclear layer is one of the important layers that we were looking at in our studies. And the length of the ONL, it's important in terms of the indication of how healthy are the photoreceptors. So the longer the ONL, the healthier and the more the photoreceptors are, and vice versa. So after the photoreceptors, we have supportive cells like the bipolar, the amacrine, and the horizontal. And those cell bodies can be found in the inner nuclear layer. And finally, we can have the ganglion cells. So those are found in the ganglion cell layer. Their bodies are found in the ganglion cell layer. And their axons can actually add up and form the optic nerve that goes into the brain. So this is a vertical section of a mouse retina, and it's something similar to the work that we do, where you can actually see the nuclei of the photoreceptors, and this is in the outer nuclear layer, the nuclei of the supportive cells in the inner nuclear layer, and also the nuclei of the ganglion cells. So you can see the three main layers that are stained through a nuclear dye. So the length of the ONL is actually important in order to indicate whether you have degeneration of the retina due to either aging or due to a specific pathology. Now, the next slide is a histological section of the human retina. So again, you can see the same layers. This is the other way around, actually. So the choroid is at the bottom in this case. So from the choroid, where you can see blood vessels, moving to the RPE, the retinal pigment epithelium, you have the photoreceptors nuclei, the inner nuclear layer, where you have your supportive cells, and the layer of the ganglion cells, and then the nerve fiber layer, which leads to the optic nerve. So this is a different type of technique, a different type of staining, but it shows the same basically information as the previous slide, but in different way. Now, let's consider, since we covered the basic information about the eye and the histology, how vision loss is associated with aging. So, in individuals who are healthy, who do not have an underlying pathology, we have a decrease in the visual function by the age of 60 years of age. So by the time that someone is up to 80 or 85 years of age, the visual acuity can actually be reduced by almost 50%. So even if someone has not any underlying pathology, the visual acuity can be minimized up to 50% at this age. And what happens in terms of the changes in vision, the age-associated changes? We can have a reduction in the visual acuity, as is seen with the best corrected visual acuity, a reduction in the color perception, in dark adaptation sensitivity, and also a contrast sensitivity. So that's related to aging and vision loss. Now let's just consider some ocular pathologies that are most commonly associated with aging. So we have the leading causes of blindness or low vision in the States and are primarily related to aging. So those are age-related macular degeneration, or AMD as I will call it, cataracts, diabetic retinopathy, and also glaucoma. So those are the main causes of low vision or blindness in the States. And according to CDC, we have more than 4.2 million Americans aged 40 years or older that are either legally blind, so that means that their visual acuity is 6/60 or worse, or they have low vision. So low vision, that means that their BCVA is less than 6/12. So this figure actually shows you how common vision loss is, even at the age of 40 years of age. So among the ocular pathologies, one of the most common one is age-related macular degeneration. So I'm going to go through some basic information with regards to this type of condition, the different stages, the diagnosis, and some of the investigations that you can do, and eventually what are the options with regards to the management. So what do we know about AMD? This is a progressive maculopathy and is associated with aging, and it results in damaging sharp and central vision. So the main characteristics in this type of condition is drusen formation, as you can see from the image that I have here from the fundus photography. You can see that the drusen is those yellow spots that you can see, and basically those are lipid and protein waste products that can accumulate in the back of the eye. You can also have pigmentary changes in the macula, and at the later stage, you might observe geographic atrophy or neovascularization of the choriocapillaries with exudation. So I'm going to go through a little bit more about the later stage in a minute. So in terms of the clinical stages, we have an early and an intermediate stage. So in this case, we do not have a significant vision loss, or we have near normal vision. And at the later stage of AMD, we have an evident vision loss, and mainly it affects the central vision. So in terms of the late AMD, there are two forms. So the first form is called the geographic atrophy, or it's also known as dry AMD. And the second form is called neovascular or wet AMD. So in this case, you can have choroidal neovascularization present. So that means that you have the formation of new blood vessels in the choroid, but those vessels are not normal in terms of their morphology and structure. So therefore, they can leak, and they can leak fluid into the retina. So how, what is the pathogenesis of AMD? We don't really know. We don't know an exact cause that is related with the AMD, but what we do know is that we have a combination of heredity and also environmental factors that include smoking and diet. So this condition is actually progressive in nature, and it increases as we age. So in terms of investigations now, a patient might start complaining that they have abnormal vision. So a simple test to perform in order to check whether we have this type of abnormality or pathology is the Amsler's grid. So I'm going to show you in a minute how that looks like, but basically you can have distortion of the vision, and this can represent active areas of macular exudation, or you can have dark areas at the grid that can represent atrophic areas of the macula. Now, if we're having a later stage of AMD and we have suspicion that we can have subretinal or intraretinal fluid, we can perform the optical coherence tomography. So this is OCT, which is a specialized test that it can show you the structure of the retina, and you can actually see whether you have fluid or not. So I'm going to show you in a minute how those looks like. Now, in terms of the Amsler's grid, this is what it looks like in a healthy, normal individual. So basically, you don't see any distortion of the grid. Whereas in case of late AMD, you can see this type of distortion in the central vision, or you can see dark areas on the grid. Now, in terms of the OCT, in the top image here, you can see a normal OCT in a patient who do not have any pathology. And you can see the layers of cells that I showed you earlier in the histological slide, starting from the chorion and going upwards. In the next set of images, in the bottom two, you can see the fundus photography, but also the OCT, and you can see that this is an atrophic macular degeneration patient where you are losing the normal structure of the retinal layers. You can also have thickness of loss of thickness of the layers of the retina, and depending on the stage, you can see different clinical presentation. Now, in terms of the management, the treatment options of AMD will depend on the stage. So the aim of the management approaches is actually to delay the progression of the intermediate AMD to the late AMD. So if we consider the early and the intermediate stages, we do not have an effective treatment yet. But what we can do is to actually modify some of the risk factors, for example, to stop smoking, to change our diet, and also to reduce the atherosclerotic risk factors also. And also, there are some studies and systematic reviews that have concluded that the administration of supplements like antioxidants, vitamin C and E, beta-carotene, zinc, and omega-3, they do have an effect in terms of protecting or delaying the progression of an earlier stage of AMD to a later stage. So this is where the omega-3 come along, and I'm going to go through a little bit more about them in a minute. In terms of the late stage of AMD, remember we have the two forms, the atrophic AMD and the neovascular AMD. So for the atrophic AMD, we do not have a current treatment. So there is nothing to be effective yet, apart from the points that we consider for the first two stages. So modification of risk factors and supplements. In terms of the neovascular AMD, so that means in case where you have choroidal neovascularization present, we do have some treatment options. So the first line treatment for this type of condition is the intravitreal injections of anti-VEGF drugs. So this is straight into the eye, injection straight into the eye. They can be quite painful and they're quite expensive also. And other options that can have an effect on the choroidal neovascularization relates to laser photocoagulation and photodynamic therapy. All right, so before I move to the omega-3, I'm going to just briefly give you an overview of a Stargardt disease, because I think this is a pathology, although it's quite rare, it's not so common as with AMD. It happens in young individuals, and it's something that is disabling at a very young age. So what do we know about Stargardt? This is an inherited condition. It causes retinal degeneration, and eventually it can lead to visual central loss and also to blindness. It's often called juvenile macular degeneration. So that means that it can mimic some of the presentation, some of the clinical presentation of AMD, but the difference is that not only it's inherited, but also the onset of the disease is quite early. So it can happen in young individuals of age 10 to 20 years of age. So this is the leading cause of childhood blindness, and the prevalence rate is estimated to be about 50 to 80,000 patients in the US. So you can imagine that the young individuals with such a condition might exhibit severe depression, among with other serious comorbidities. So it can have a negative impact on the quality of life of these young patients. And it is essential to note that, again, there is no treatment available for this condition. Although the research is being done for some type of gene therapy or some stem cell therapy, but still there is no official treatment for this type of condition. So this is where the omega-3 come along the way. So what do we know about them? Let's think about these type of fatty acids first before we see how they correlate to some of their therapeutic potential in eye pathologies. So in terms of polyunsaturated fatty acids, we have two major types. So the first one is omega-3 and the second one is omega-6 fatty acids. So there are quite a few of these fatty acids, but the main scientific focus is on three major omega-3. So the first one is alpha linoleic acid, so ALA. The second one is eicosapentaenoic acid, APA, and the third one is docosahexaenoic acid, DHA. And also we have a major omega-6 fatty acid, which is related to arachidonic acid, AA. So the fact that these omega-3 cannot be synthesized in our body they are considered essential fatty acids. So that means that we have to obtain them from our diet. Otherwise, it's quite hard for the body to synthesize them. Although you can synthesize some of the EPA and DHA from the ALA, but those are very insignificant quantities. So ALA is present in plant oils, for example, some nuts, flax seeds, soybeans, and DHA and EPA are usually found in fish oils like salmon, sardines, and so on. Omega-3 are important in terms of cell membrane structure because they are components of the phospholipid membrane. And DHA is specifically high in the retina, but also in the brain. So it's important here to note that because we have a lot of DHA in the retina, it's another point to consider is also EPA. So we need to find a way to incorporate EPA into the retina in order to see a protective effect from these fatty acids. Now, in terms of weighing the balance between the omega-3 versus the omega-6, let's see how this is related to the eicosanoids and the eicosanoids pathway. So in addition to the energy provision from the omega-3, we also can form eicosanoids. So what are the eicosanoids? Let's just remind ourselves. So the eicosanoids, they have a wide range of function, and usually they have an effect on the cardiovascular, on the pulmonary, on the immune, and on the endocrine system. So these, the eicosanoids that are made from the omega-6, for example, from arachidonic acid, which is a major omega-6, are generally more potent mediators of inflammation. They can also cause vasoconstriction, and they can induce platelet aggregation compared to those eicosanoids that are made from omega-3. And interestingly, EPA can actually compete with AA for the synthesis of the eicosanoids. So that means that if you have a high amount of EPA in DHA compared to AA, you will shift the eicosanoid balance towards a less inflammatory activity. So this is the desirable effect that you want to see. So if we take a look at the diagram here, you can see that from the omega-6, like the arachidonic acid, you can form pro-inflammatory mediators like prostaglandins and leukotrienes. And from the omega-3, like EPA and DHA, you can have anti-inflammatory mediators like the resolvins and the protectins, but you can also compete with AA in order to prevent formation of pro-inflammatory mediators. So that brings us to the significance of the balance between the omega-6 and the omega-3 in the progression or the resolution of an inflammatory response. And this is the point that I would like to stress on, that there are quite a lot of studies that have checked the ratio between the omega-6 and the omega-3 fatty acids. And they have found that the higher the ratio of omega-6 versus omega-3, the more likely the progression of a disease like a cardiovascular disease or cancer is. And more specifically, since we're talking about eye pathologies, they also found this in different eye pathologies, whether this was done in preclinical models or in clinical trials. Now, let's relate omega-3 fatty acids with ocular pathologies. There were numerous studies that were performed in terms of evaluating the efficacy of omega-3 in preclinical settings against retinal damage. And this was reviewed by San Giovanni in different pathologies of the eyes that were related to inflammation, to ischemia, to light, and to oxygen. In terms of clinical trials, though, we have inconsistent data of the effect of omega-3 in eye conditions. And the reason for this is, I believe it's because we don't have consistency in terms of the actual dosage of this omega-3. So we hypothesize that if we have if we administer omega-3 fatty acids daily at a high enough dosage, in a way that these fatty acids can get incorporated into the retina, we will see a therapeutic effect. So this is the hypothesis that we had in our mind. So that brings us to the main aim of the studies that we have performed so far, which is to investigate the effects of daily oral administration of omega-3 fatty acids in either healthy aged animals or in animals with an underlying ocular pathology, keeping in mind to maintain a ratio of omega-6 to omega-3 at about 1 to 1.5. So the ratio that we were looking in particular was the AA over the EPA. And the reason why we try to maintain this type of ratio is because we did some preliminary studies before we actually performed the efficacy studies. And we have seen that when we have this type of ratio, when the ratio is quite low, we do have a significant increase of EPA in the retina. Now, in terms of the preclinical studies that were performed by our research team, we did investigate and confirm the effects of omega-3 in a variety of animal models, including normal aging, of a model of retinal degeneration, of Stargardt disease, of optic atrophy, of anterior ischemic optic neuropathy, glaucoma, and the last and most recent study was performed in retinitis pigmentosa model, which this result has not been published yet. So what I will do today is actually going to cover the first two studies, preclinical studies that were performed in our group, and because I don't have the time to go through the whole all the studies that we performed so far.
Actually, Katerina, we have about 10 minutes, so we can allow students to ask questions.
Okay. So in terms of the preclinical studies, I have the normal aging model, where in this case, we did actually see that we have protection of the retina from the age-associated degeneration in the black 6 mice. So what happened here is where we have here three groups. One of the young black 6, they were untreated. Another group where we have aged black 6 that were untreated, so they were 24 months. And in this case, the third group included aged marched, so that means they were 24 months of age, and they were treated with omega-3, mainly with EPA in this type of formulation and some DHA. Now, in terms of the study design, we have administered oral omega-3 for 60 days, and that was done daily. And we had obtained blood samples on day 0, day 30, and day 60 in order to monitor the AA to EPA ratio. Now, on day 60, which was the end of the study, we have prepared the eyes for analysis. So what we did was we checked the fatty acids in blood samples, but also we have isolated retinas and we have checked the fatty acids with the use of gas chromatography. With the use of electron microscopy, we have checked Lipofuscin granules and we have quantified them. So Lipofuscin granules are waste products basically that relate to the progression of retinal degeneration. And then we have looked at the photoreceptor outer nuclear layer through light microscopy. And finally here we performed some mass spectrometry-based proteomic analysis of isolated retinas to identify differences in proteins. So what we confirmed is that we've seen that the AA-EPA ratio was indeed lower after the day 30 and day 60 of treatment. And we have also seen that EPA in the retinas on day 60 was also increased in the treated group. So that showed that treatment can actually increase the EPA in the retina. So this is something that it was desired. The next set of results shows the Lipofuscin granules. So these are images from electron microscopy in young, in aged untreated, and aged treated mice. And as you can see from the image, we have an accumulation of lipofuscin granules in the aged untreated mice compared to the treatment group. So within the treatment group, we have seen significant reduction of the lipofuscins. Now, that means that less lipofuscins, less progression of the disease, meaning more protection of the photoreceptors. So this is the most important part of the study, I think, where you're looking at the outer nuclear layer, measuring the length of the ONL, as indicated by the white line. And in this case, you can see that young individuals have about 200 micrometers thickness of the ONL, whereas the untreated aged animals had a much lower, a much shorter ONL. Now, if you compare it to the treatment, you can see that there is a significant increase of the treatment compared to the untreated group. So that shows you that treatment with omega-3 indeed protect the photoreceptors. The next set of data relates to the proteomic analysis. So in this case, we have found very interesting results that relate to myelin-related proteins. So basically, as you can see, the omega-3 treated group showed an increase in the myelin basic protein, in the myelin proteolipid protein, and also in the myelin regulatory factor-like protein. So in all cases, we've seen an increase in that. And that shows that perhaps there is a mechanism that relates to myelin synthesis, and it might indicate that there is a possible regenerative mechanism with the use of the omega-3. The next set of the results relates to the retinal degeneration model. So I would like just to briefly go through some set of data. I'm not going to go through the whole thing and show you again what happened with regards to the study. So very similar study design as the previous one. In this case, we had a group with omega-3 plus omega-6 and a group just with omega-3, in addition to the control groups that we had. We have treated the animals for 90 days, and on day 90, we have excised the eyes for preparation. We have looked for fatty acids in blood and retinas, and we have looked at the photoreceptors outer nuclear layer, in this case with confocal microscopy. And in addition, we looked at different inflammatory markers at the gene level through RT-PCR, but also at the protein level through Western blot analysis. So what is interesting here is that if you take a look at the images, you can see it's evident that from image D, which is treatment with omega-3 only, and as you can see from the graph also, you have a large increase in the ONL. So that means that you have more photoreceptors after treatment with omega-3 compared to treatment with omega-3 plus omega-6. So the greater protection is seen when you treat the animals with omega-3 only. In terms of the inflammatory markers, I'm only going to mention the Western results at the protein level. So we've looked at isolated retinals and checked for specific inflammatory markers like TNF-alpha, interleukin-17, and interleukin-18. But the most evident effect here was that after treatment with omega-3 only, you see a dramatic decrease of this inflammatory marker, which perhaps is related to the pathogenesis of retinal degeneration in that model. So therefore, reduced inflammatory process through the effects of interleukin-18. Now, the next studies included very similar study design. So this was performed in a model of Stargardt disease. This one related to the effect of omega-3 in a case of optic atrophy. The next one related to a model of anterior ischemic optic neuropathy, where we've seen the neuroprotective effects of the omega-3. And the last one here was a study on glaucoma model. So all of these studies showed the same thing. So they confirmed that omega-3 do work. So what is the conclusion that we can draw from these studies? Basically, you need to eat more fish. That's the important point. So long-term daily oral administration of omega-3 fatty acids at an adequate dose, when you can actually incorporate EPA into the retina, can protect the photoreceptors in either aged healthy mice or pathological eyes through different mechanisms. Now, if we can translate this in a clinical setting, that means that you can minimize the progression of vision loss, or you can even improve it. And just a brief account on the current study that we have completed recently, and we do have the data analyzed. This was a study where it was designed as a prospective multi-center double-blind placebo-controlled trial. The patients were recruited in France and Italy. We had patients with moderate or severe dry AMD or Stargardt disease. We have treated the patients with mainly EPA and some DHA for 6 months, and during that period, they had visits at the ophthalmologist, and we performed different tests. And basically, we do have promising results that show that omega-3 administration can improve vision, and it can prevent further vision loss after 6 months of treatment. In terms of future studies now, my plans are to investigate whether omega-3 do have regenerative potential. I think this is something quite important. If we demonstrate that there is cell division in the photoreceptor layer, we would like to design and to develop a genetic animal model tool in order to describe this type of mechanism behind the regenerative potential, if it does exist, and also to identify which retinal cells might serve as stem cells that can signal cell division of the photoreceptors. In terms of clinical studies, we would like to perform a larger scale multi-center trial, perhaps in the US, that we can include patients with AMD or Stargardt disease again. But in this case, we want to divide the patient according to their pathology, according to the severity, and also perhaps to have different doses of the treatment, or to perform preventative studies in order to see whether at an early stage you can avoid the progression to a later stage of the disease. And before I finish off, I just wanted to thank Ophthalmos Research and Educational Institute for giving the opportunity to perform all of these studies and for funding these studies. And also I have a lot of collaborators in the Cyprus Institute of Neurology and Genetics. And I'm not going to thank everyone individually, but I couldn't have done all of this work without them. So thank you. And the take-home message is that I hope I convince you to eat more fish and take some of the omega-3 because they are good for your eyes.